Good morning, good evening. My name is Akira Onoda from NGENIESS Biosciences. I would like to introduce our N-link technology for protein-based drug. We will revolutionize protein therapeutics through advanced bioconjugation technology. So here's our technology. So if you have a protein of interest and you modify other molecule to the target protein, so our technology enables the one-step conversion to the conjugation regions, named TFOC. With this region, the protein of interest can be modified to this protein, which has specifically N-terminal modification. This is the simplest and specific bioconjugation. So we're convinced this is a really powerful technology, so we are now trying to apply this conjugation methods to the protein-based drugs. So of course, the cancer treatment is a big problem for the many pharmaceutical company make drugs, and particularly lung and cancers have a lower 5-year survival rate. So the pharma company produces many types of drugs, and then the powerful candidate is an antibody-drug conjugate. So an antibody-drug conjugate consists of antibody that specifically recognize the cancer cells, and the drugs that kill the cancer cells. These two components are conjugated. So the ADC specifically delivers the drugs to the cancer cells, and the normal cell does not kill in this treatment. So because this ADC is really powerful for the cancer treatment in nowadays, so the big pharma is extensively developing these ADCs. So there's a couple problems in ADC development. So the first, the high cost in production. The second, it takes a long period to optimization. And three, so bioconjugation method is limited for the ADCs because there's many licenses for the linker technology, which is really important for the ADC. So there's a great need for the new bioconjugation method for the ADCs. So bioconjugation for ADC needs to be required in terms of specific numbers because the drug antibody ratio, DAR, should be specific. It should be fixed. Also, the modification position should be specific. If not, so the ADC should be randomly modified and also be inhomogeneous. So these are the first generations of ADC, still also in the market. And the current technology enables specific modification, and homogeneous ADC is available. So with the conjugation, our N-link technology for the other modifications should be implemented in the ADCs. So this is really a great advantage in the clinical aspect. So the ADC is evaluated as therapeutic index, which is defined in the ranges between the maximum tolerated dose and the minimum effective dose. So if you can have ADC with a specific modifications, the therapeutic index is widened. So this is very good performance and a good efficacy. But with the conjugation, our N-link technology, so the new ADCs would have wider therapeutic index. Also, this would prolong blood circulation or control membrane association. So new other properties could be implemented in using our N-link technology. If to do this, so for example, peptides, proteins, nucleic acids, variety of molecules could be very useful to conjugate with the antibodies. With our technology, this can be enabled by only 2 steps. Again, the simplest in specific fashions. So our technology will give the solution for the problems in ADC section in pharma. The first, with this technology, production costs could be reduced. Also, the optimization period could be shortened because our modification technology is very simple. Also, furthermore, our technology would give added opportunity for the new attachment linker. This would enable to generate the new type of ADCs. So global market for ADC is really growing. In 2027, it will be expected to grow 18.4 billion. So we targeting the 40 million in this market. So we will do the business for the get business for the license fees with our collaborators who will develop the drugs using our technology. Also, we will add more business revenues by selling our precursor molecule that will use for the ADC drugs, who is interested in our who is produced and then interested in our technology. So here's competition. So of course, there's a lot of ADC or linker technology is developed in biotech and pharma companies, but all these companies using a lysine or cysteine conjugations, which this is different in our, we use the N-terminus, which is pretty different in the other companies. So this our modification enables the simplicity and the specificity. In addition, our technology is also really applicable for drug delivery systems because our cleavable linker will generate intact protein, which is quite unique in the section of those conjugation methods. But also, the important thing is this conjugation method is also comparable with the other technologies. So I think the competition, these are the companies for the possible collaborators. The application of this is not in medicine, but also diagnosis by materials. So we have a lot of variations of combinations of the proteins and conjugation. We have already 2 companies started partnering and started collaborations. So, and we are building still building a team, and I will finding a CEO and a CMO for the future teams. Here's a development roadmap. In 2023, we will finish the proof of concept experiment, and for that we need $3 million. So thank you for your kind attention.